Hey, welcome to the Bioinformatics Chat. Today I'm talking to Jennifer Liu. Hi, Jennifer. Welcome to the podcast.
Hi. Thank you for including me on this.
Could you tell us a bit about yourself?
So, I am a PhD candidate at Johns Hopkins University. I work with Dr. Steven Salzberg, who has worked on a number of genome assembly projects in the past, and mainly we're a computational lab that develops software for analyzing various genomic data sets.
So your background is in computing or biology?
I actually have a bachelor's degree in chemical and biomolecular engineering from Johns Hopkins and a minor in computer science. So when I wanted to start my PhD, I really wanted to combine the biology background and computer background that I had and kind of put it towards, some interesting project in the biomedical fields, yeah.
So we're going to talk about Bracken, which is one of your works, but I noticed that since then you worked on very different projects not related to metagenomics. Is that right? What's sort of the vision of your PhD thesis?
So I don't have a concrete PhD thesis, but generally all of my projects revolve around the idea that we can use all of the genetic information that we have right now, so all of the different genomes that are already assembled and finished, and use those to help diagnose human infections. And so a lot of it is relating to metagenomic data sets, microbiome data sets specifically, in that we're trying to be able to sequence, an infection from any patient and try to figure out what they have using the software that we have. And so Bracken was kind of the next step from Kraken, which is one of the main pieces of software that we use to try to identify what is causing this infection, what is in that sample that we got. Because essentially the steps that we have from patient to, when it comes to my computer, is we get the sample from the patient, we try to amplify, extract any of the DNA from that patient, sequence it, and then all of that data from the shotgun sequencing goes to us, and we run Kraken and Centrifuge and Bracken to try to figure out what is actually affecting and infecting the patient.
So do you collaborate closely with a hospital?
Well, we have a number of collaborators at Johns Hopkins Hospital, and then within even the Johns Hopkins network, we have a number of pathologists that are also part of this project, a couple other professors, and then a couple other clinical doctors that have patients that are currently or have in the past been infected with either unknown or known pathogens. And so that's basically where all of my projects kind of revolve around. And so Bracken was just the first project of mine, and it was just this is one part of our software puzzle that needs to be solved, and so that's what I focused on first, and then I moved on to more just databases and having the genomes themselves.
Yeah, interesting. So I want to talk about Bracken, which is your tool, but in order to understand that, I think it's crucial to also understand Kraken, which is sort of built upon. And so I recorded a podcast, I think it was episode number 7, which listeners could go to. But can you remind briefly what Kraken is and how it works, and why it's not enough by itself? Why is there a need for Bracken?
So Kraken is a classification tool developed by Derek Wood, who was a PhD student in Steven's lab. And essentially, at the time of the development of Kraken, shotgun sequencing was becoming more and more popular. And so there was just an abundance of DNA. So I think a lot of people already know about BLAST, which is a great tool where you have a few genomic sequences that you're trying to classify and figure out what they belong to. But when you have a data set when you sequence with Illumina or any other sequencer, you end up with possibly billions of DNA sequences, and running blasts for all of them is just unfeasible. And so Kraken was actually developed to be a sequence classification tool to try to take all of that data and really give you a comprehensive overview of like what all of those sequences are. And so it's a different technique to some of the other ones out there, but it essentially works on exact k-mer matches. The default is 31 mers, where it looks for it takes every sequence that you're given, every sequence in your data set, and it looks for exact 31 mers that match their database to oversimplify what the algorithm actually is. But it also identifies the fact that there's a lot of genomic sequences out there that are identical across genomes, across species, even across genuses. And so instead of trying to arbitrarily choose which species a sequence might belong to, even though it might be identical across, like that 100 base pairs might be identical across different species, it'll give the lowest common ancestor. So a lot of people already use Kraken for their classification work, but I think the reason why we decided to make Bracken is we actually found that a number of people were assuming that Kraken was giving you species abundance numbers. And as a result, because Kraken just doesn't even try to push everything to the species level, but gives you the lowest common ancestor of what it's certain that read belongs to, we decided to make Bracken because Kraken's just going to strand reads at higher taxonomic levels just on the basis of what the classification algorithm does. And on the basis that if you have a 100 base pair read that is identical across different species, Kraken wants to give you the most accurate classification that it can. And so it's just going to put it at the genus level, for example. But because people really wanted to use Kraken's data to do species abundance, that's when we decided to make Bracken. And I think that something to emphasize is that Bracken is not going to give you exact species abundance. But the reason why we say that it's estimating the species abundance is because you really can't tell necessarily. If you have 100 base pairs that's identical between two different species, you really can't tell exactly which species it came from. Not in the way that our algorithm works, and not in just like general. If you know that this is 100 base pairs that's identical between two species, I don't think anyone would be able to tell you which species it came from. So we actually created Bracken to be a more probabilistic method, where it's trying to help you identify where that, or estimate where the reads might have originated from.
Yeah, so the core, not sort of deficiency, but maybe the core thing that Kraken doesn't do is assigning sequence reads to the species level, right? Because if it assigned sequence reads to the species level, you could just count them and normalize, and you would get roughly the abundance. Right. But this is where I'm a bit confused, because when I'm reading the Kraken paper, it tells me that it builds this tree, the taxonomic tree, and then it assigns each k-mer to the to like various nodes of the taxonomic tree. But then when it classifies the whole sequence, which consists of multiple k-mers, it finds the leaf in the tree which has the maximum root to leaf. So if you construct the path from the root of the taxonomic tree to the leaf, and you sum up the scores along that path, so it finds the leaf which has the maximum sum of scores. And right. I don't quite see how you could end up at the genus level. Because genus is not a leaf, right? And it looks for the maximum root to leaf path.
Well, so the tree that we're talking about, so it constructs this scoring tree for every read. So it doesn't construct the scoring tree for all of the reads. And so I think the thing to note that is if you have, I'm just going with the arbitrary 100 base pairs, 100 base pair read, and every single k-mer within that read can only be classified as specifically at the genus level, it's not going to give you a leaf classification. The leaf is going to be that genus. So the way that the database is created is that every single K-mer from the entire database, it's given a specific taxonomic ID based on like where it falls in the tree. And so when a single reed is being classified, every single K-mer is put in that tree. But if every single K-mer in a single reed is going to be at a higher taxonomic node, then it's just going to get that classification. Does that make more sense?
Yeah, absolutely. That was my confusion. So we're not looking at the taxonomic tree, but we're looking at the sort of reduced tree, which only has the nodes that it found.
Right.
Among the K-mers. Yeah. But still, do you have any statistics, like in realistic example, how many, like what percentage of reeds get mapped to, not to the species level? Because I'm thinking in order for a reed to be mapped at the genus level, it has to be like very exact sequence. I suppose there is a fair bit of homology within a genus, but still, even like a smallest variation will pinpoint it to maybe a species level, because if at least one K-mer in that reed gets sort of pushed down to the species level, that sort of destroys your equilibrium, and it will never get back to the genus level. Does that make sense?
Right, yeah. No, I understand. So I think that the thing to note is that we're doing all of this based off of the taxonomy that NCBI provides. And so even within that taxonomy, I would say that there's a number of genuses in which the species are highly related. And so we use a couple of those examples even in our Bracken paper, where we talk about the fact that different bacillus or different mycobacterium species are highly similar, like nearly 99% similar. I don't remember the exact numbers, but because of that, like most, if not all of most of the K-mers from those species are at the genus level. And so unless you get 100 base pair reed that somehow captures that 1% that is unique to that species, all of the K-mers from that genome are going to end up at the genus level.
Right. But do you have a sense of like very ballpark figures? Like if we took a typical metagenomic sample, it would try to classify it with Kraken. What percentage roughly of reads would be at the species level, like genus level, family level? Do you have a sense of those figures?
I don't have them off the top of my head. I know that our lab has run a number of samples through both Kraken and Bracken, and I don't have the numbers off the top of my head, but I will say that even like a few hundred reads out of like maybe 40 billion always end up even just at the bacteria level. In that like the K-mers from those few hundred reads can't be classified anymore specifically because they're shared across bacteria that are not at all related to one another.
Am I correct that sort of the push for creating Bracken was this paper from Lior Pachter's lab where they applied Callisto, which is normally an RNA-seq abundance estimation tool, and they applied this tool to a metagenomic sample, and they demonstrated that it performs better than Kraken. But then, of course, they ignored this fact that Kraken can classify some sequences at the higher taxonomic levels.
So admittedly, that was the paper that kind of showed us that if people wanted to use Kraken as an abundance estimation tool, they were using it incorrectly. And so we wanted to provide the people that use Kraken already with something that can allow them to do that abundance estimation. So yes, that was the bioarchive paper archive paper that we noticed prior to the development of Bracken.
Right. So you noticed that people are not using Kraken correctly. And by the way, what is the correct usage of Kraken? So before Bracken was written, right? So there was no like easily available way to transform Kraken classifications into the abundance data. What was the intended way to use Kraken? What sort of information could you get out of it?
I mean, very simply, it's a classification tool. So it's not meant to... It does help you kind of summarize what's in a data set, but not like at a specific taxonomic level. I think for a lot of data sets, even for the ones we use, we always look at the Kraken classifications first, because I think that that provides the most information. I think, as you said, reads at the higher taxonomic levels, it's not as useful. I think in our purposes, and even in some other people's purposes, what's most interesting is what's at the genus or the species level. And so we still really care about like the number of reads that we can see like across the species to genus and so on and so forth. And I think that that's where Bracken has the most impact if someone wants to do abundance estimation at a very specific level. So I think for Kraken users, it's a really good way to summarize what's in your data set. And I think that like having the different levels is also like really informative. That's what makes Kraken's accuracy so high, is because it doesn't try to assume that it's definitely from a different level, or it's definitely at the species level, rather. And so I think they have two... I think both Kraken and Bracken classification and estimations are really useful. But I think in terms of just like how you summarize your data set, Bracken is probably an addition and a welcome addition to help you summarize your data set at a species or a genus level, which some people aim to do in their papers or when they're just trying to summarize broadly across a data set of theirs.
Yeah, definitely. So Bracken helps a lot, but I'm trying to understand if you only have Kraken, and at the time you only had Kraken, right?
Right.
So you're saying it's a way to summarize, but it classifies. So as you said, we have 1 billion sequences, and if we classified each one of them, that's not really a summary. You cannot easily look at the 1 billion taxonomic labels and tell anything, right? So one way to summarize this data would be just have a set, like ignore all the counts, ignore all the specific sequences, just gather a set of taxonomic labels that were used, and that sort of gives you an idea of what is in there. But I imagine it's very fragile because even if a single read was classified at some odd species, then the species gets into the set. But then you're saying that if we were to use this properly, if we were to use Kraken properly, we should ignore all the count data because we cannot really summarize the count. The count data is meaningless in Kraken until you apply Bracken. So you cannot look at the counts. Like I imagine if your aim was to combine just a set of taxonomic labels, you could do that faster, and you don't need much precision if you don't care about each particular sequence. You just care about like what's in there.
So what I'm trying to say is that for Kraken, it wouldn't be useful to try to summarize your data in percentages. I think the read numbers are really important and really interesting to look at. So a lot of times when we look at our data sets through the Kraken reports, or even when we're just trying to understand whether a read is truly classified at the right taxonomic ID, what we really are interested in is we first look at the reports and we try to see like the distribution of numbers. So like how many reads ended up at like this genus level, or how many reads were very specifically at a species level, and so on and so forth. And then from that, we can kind of go back to the full read report where we can say like, okay, this read was classified at this species level, but like if we blast that read, what do we get? Because I think that something to note is that Kraken is, as long as one k-mer within that read matches exactly, it's going to give you a classification. But the databases that you use for Kraken isn't necessarily going to have everything in the entire world. And so what we want to do is we want to both confirm just to make sure that that's what that read is meant to be, and then also just like what Kraken really does, and what it does really nicely is it gives you a classification for every single read. And so we can kind of go back and look at the read and be like, oh, is this a low complexity sequence? Is this a high quality sequence? And just do more analysis on the read level. Whereas Bracken isn't going to give you a per read classification.
Yeah, that's a good point. So if I understand correctly what you're saying, we could use Kraken in order to find some interesting cases, maybe the ones we didn't expect. And then since it tells us which sequences it classified this way, we can sort of zoom in on them and go from there.
Yeah, and Bracken is much more of an overview kind of program, so it's not where you're like looking at specific reads classified as different species, but it's more when you just like want to know as a summary what's in your data set.
Yeah.
Yeah.
Okay, so Bracken sounds like a great tool, but how does it work?
So we tried a number of iterations for Bracken, but essentially we didn't want to start from scratch. We didn't want to create a new abundance estimation tool, but we really wanted to provide something that people that use Kraken could use it very simply. And so what we do is for every classification that Kraken gives, Bracken is essentially calculating a Bayesian term, basically a probability of where those reads might end up. So, for example, for all the reads that end up at the Mycobacterium genus level, Bracken is going to tell you what's the probability that those reads belong at MTB or M. bovis, and it's based on those probabilities going to redistribute all of the reads at the genus level down to the species level. That's the broad spectrums of what it does. And I guess we use Bayes' theorem because we found that this is the most accurate way to really make use of both what we know in terms of how those species might be similar or unique and kind of compare them to one another. So we use a lot of prior information in terms of what's in your database to determine like how reads from those databases might end up being distributed.
Yeah, so the way I found most useful to look at this while I was reading the paper is as a two-step estimation. So this is a bit contrary to what people usually understand by Bayesian analysis, because usually you have some priors, and then the data informs your likelihood, and then from there you get your posterior probability. But in your case, you estimate both the prior probability and the likelihood from the same data, but just looked at a different, or I guess you partition your data, right? So you look at the uniquely mapped reads, and that informs your prior, and then you use that prior to look at the reads that were sort of ambiguously mapped, right? And then that allows you to redistribute those ambiguous reads.
Right. So the first step before you do any data set is to basically try to figure out how, if you broke up your entire database into 100 base pair reads, like where would they end up in the tree? And then use that information to then figure out, okay, then what's the probability that these reads ended up here because they originated from this specific species.
And so this reminds me a lot, again, what's going on in RNA-seq data analysis, and there people usually apply the expectation maximization algorithm, which is essentially these two steps but taken further. So we can apply the same step over and over again, right? Once we redistributed our reads, we can use that as our new prior,
right?
And basically repeat the procedure, and we may get like slightly different numbers. And so you repeat that until it converges sufficiently, I guess. So did you consider using that, or was there a reason you're doing just one iteration of what essentially is an EM algorithm?
So I did consider whether repeating it would be useful, but I think we tried this with a number of data sets and a number of simulated data sets in our databases. And I think what we found is that while we could repeat this process, I think that in just the one step, I think it was sufficient for what we were looking for. So I do remember like reading a number of papers where EM was, expectation maximization was their main part of how they did all of the species abundance algorithms. But I think for me, what I really wanted is what I wanted was a program that would be really useful for people that were using Kraken, but also sufficiently and I guess accurate to a degree. And so we tried a number of different ways, and I based, even just with the one EM step, as you called it, the accuracy was actually really high. The number of reads that were off was really low. And so we calculated a number of different, I guess, methods of figuring out the accuracy. So we calculated a number of different numbers that would tell us like how accurate that algorithm is. And even with the one step, Bracken was coming really close to what it was. And I think that like something to note is that we're also basing this off of Kraken classifications, and Kraken itself is already very accurate, and we didn't want the numbers to stray too much from what Kraken was already giving us. And I think that recalculating everything would kind of go beyond what Kraken has already done for us. And Kraken's already giving you a lot of information by telling you kind of where all of the reads originated from. And so I think that to do more EM steps would kind of be ignoring the information that Kraken has already given us in terms of, and then Kraken already has very high accuracy.
Well, you wouldn't exactly be ignoring anything, right? You'd be essentially using the same data, just driving your algorithm to convergence, as opposed to like stopping. And it may very well be that one step is enough, if you say so.
Yeah, I do think that, so what we did try to test is, in one of the steps, as you said, we calculate like how unique each genome is. And then we actually did try a step where instead of using the full algorithm, we instead used the uniqueness of each genome and then the number of reads very specifically classified at that genome to kind of scale up in a sense, to see if that would actually be accurate. And what we found is that, like, it's highly sensitive to the number of reads that were actually classified at that specific species. Because we're running under the assumption that, like, every read from a genome is, like, in your data set. You're running under the assumption that the full genome was captured by all of your reads. And so when we actually tried that just simple scale up, we found that it definitely overestimated and underestimated for different genomes by a lot. The accuracy was far lower. And so that's why we turned instead to this, because we're trying to make use of just like what's within that particular tree. So we didn't want to recalculate for the entire tree what everything was, but we really just wanted to focus in on that tree and keep those reads within that section.
We started to talk about accuracy, and did you try to compare Bracken to other tools like Metaflan or Callisto?
So I think we did take a look at like how Bracken would differ from those other tools, but I think that something that we really fought for, I guess, even in the paper submission process, is we didn't want this to be just another tool trying to compete or overtake any other tool out there. But what we really wanted is we wanted to provide this as an additional resource for Kraken users already. So many people already use Kraken, and this is something that we wanted to put out there as useful to them and not necessarily as saying that this is better than any other tool out there. I think that like a number of reviewers definitely wanted us to compare and include that in our paper, our program to all of the other ones out there. But I think that what we really fought for in putting this tool out there is that this isn't meant to compete with all of the other ones. This is really meant to stand on its own with people that are already using Kraken. And I think that has already been proven useful, because there's a number of people that have used Kraken that started using Bracken as well to really, for their data sets, for their analyses, and for their experiments. And that's really what I wanted. I do remember that we did run Metaflan as well, and I think we attempted to run Callisto, but we ran into problems with the software that I don't specifically remember. But because it wasn't our focus, we didn't pursue that further. And the Metaflan results were pretty similar, although we didn't compare them in terms of numbers, so I don't remember the exact numbers in terms of accuracy. I don't think that necessarily Bracken is meant to replace Metaflan, or Metaflan's better than Bracken. I'm not sure.
Yeah, so I kind of see where you're coming from, why you wouldn't want to compete with anyone, and isn't that what like we all want in our lives, just not compete with everyone and just live happily.
Yeah.
But I can also see the point of the reviewers. So even though you don't want to compete, you will essentially, like, your tool, your baby, will compete, because if we imagine a biologist with gigabytes of data, and they want to analyze the data, and they have these tools available. They have Bracken, they have Metaflat, they have Callisto, they have some other tools. So naturally, they will ask, well, what tool will perform the best for me? What tool will give, let's say, the most accurate result? Now it's out of your control once you release the software, so the people will conduct this competition for you, right? And I think it's useful, even though you didn't compare them, but we can still talk about the algorithms. And just looking at the algorithms, it seems to me that Callisto must be more accurate, because otherwise it's doing pretty much the same stuff. So it's also looking up k-mers. It's also running, I think it has a similar statistical model. I'm not sure if it's probably not exactly the same, because Callisto doesn't work off a Kraken database, but it probably has a similar statistical model. An obvious advantage that Callisto has is that it doesn't operate off this intermediate representation. So it has access to the individual k-mers. So it simply has more data available to it. Whereas in Bracken, because of the constraints that you imposed onto yourself, you have to work off the data that Kraken gives you. And that data is very reduced. Because Kraken itself, for every read, it knows where each k-mer maps, but then it collapses all the data into a single node in the tree. It just finds the maximum path in the tree, and it reports only that. So obviously you get much less data. It would be very improbable if Bracken managed to be as accurate or even more accurate than Callisto, wouldn't it?
So they did actually compare Callisto to Bracken in their final paper, which they did publish, I think recently, in July 2017, in Bioinformatics. Not that I'm trying to promote their paper. Yeah, but they did do the comparison. So I think even through reading through their paper, and even a number of people that have tested the two against one another, I do think that there are similarities in their algorithms, and Callisto does provide more information to the algorithm, but I think that that's what I would say is the main difference. Is that in theory, providing more information to the algorithm would make it more accurate, but I think it's also you provide a trade-off then to your program, in which if you provide more information to the algorithm, you're hoping for more accuracy, but you're also using up more memory. And so I think that that's the main difference of anyone that I've ever heard trying to use Callisto for such metagenomic classifications or abundance estimations, in that the memory differences between the programs is significant. So I tried reading through their paper, and I don't really know how they ran Bracken. The runtimes that they provide for Bracken seem fairly off to me, because Bracken actually runs in less than a minute, to the point where it wasn't measurable, but they claim that Bracken ran for much longer than that. And so I'm not really sure. In terms of the accuracy, it's pretty similar. In terms of the runtimes, they claim that Callisto is much faster, although I have no knowledge of how they ran my program. And so I'm not really sure where their runtimes are coming from. Bracken itself runs fairly quickly.
Yeah, isn't that a huge problem in bioinformatics, that every lab tries to show their tool in the best possible light? And so this runs a bit counter to the spirit of science, where we should be able to figure out which algorithm actually is the best one, or at least even if there is no clear winner, we should be able to say that, the scientific consensus, we have scientific consensus that smoking causes lung cancer. So in the same spirit, we should be able to say that there's a scientific consensus that this algorithm sort of consumes more memory, and this algorithm runs faster, and this algorithm produces more precise results under certain conditions. But instead, it seems to me that we're treating our tools more like salespeople, right? we sort of sweep all the downsides or deficiencies under the rug, and
I think that's why I find it hard to even compare myself, or I guess my program rather, to Callisto. And I think that that's also something that when I was talking to my professor about whether or not we should include the comparison information in the paper, I think that that's something that we definitely fought against, because I think that, yes, reviewers want to know, is this novel? Is this going to be better than what's out there? But I think for us, it's we're already biased, because this is the program that I wrote. And the people in Leo Pachter's lab are going to be biased, because Callisto is what they wrote. And so I think that, what I've found, even at conferences when people talk about the comparisons between their programs and Kraken or Bracken, all of us want to paint our own programs in the best light. And so
yeah,
when we're doing comparisons like that, we're going to be biased. And I think that it's better left to a third party to compare them instead. And I think that that's something that, I've definitely fought against, because I can tell already the competitive nature of academia in only my first couple of years as a PhD student. Whereas I would prefer if other people tested and compared and made that decision for themselves. And someone already has for Kraken, where they've determined that Kraken's accuracy and speed and memory is better than most out there. There are comparison papers for that already, and I don't want to rehash any of that. But I think that I would, even if I was a third party, I would trust a third party to make those comparisons rather than the authors themselves. I think that I also would say that every program is only as good as its database. And I think that that's why I'm focusing on databases now, because what I've also found is that you can construct a database that's going to be perfect when it's run on Callisto, but not perfect when it's run on Bracken. And even when it comes to the two programs that came out of my lab, Centrifuge and Kraken, you can construct a database that's going to work perfect on Centrifuge and not so perfect on Kraken. Not by a lot of differences, but by enough that you could say that one is more accurate than the other. But I think that what I'm focusing now on, and the reason for this, is because you want to have the most accurate database to begin with. And I think that every tool is only as good as its database, and I think that that's something that people don't really think about when they look at the different algorithms, because they're looking at what the program does and not what the program is actually operating on.
Mm-hmm. So what is the state of Kraken databases? Does your lab maintain an up-to-date version of a Kraken-compatible database that people could download?
So we have a number of Kraken databases that we personally use, but they're far bigger than what we would provide anyone with. And so something to note is that when Kraken was first released, we provided a database called Mini Kraken that people could download that was only 4 gigabytes. And it was based off of, at the time, what was in the bacteria, archaea, and viral RefSeq databases. And so it was using all of those, not with any picking or selecting of which genomes. It was using everything from those databases. And so we're working on constructing a new mini Kraken database for download again. But something that we also note is that, so the databases can get quite large depending on what you want to have in them. So Kraken is not going to be the tool that I would suggest to anyone, and Bracken definitely not, for someone that wants to be running everything against the NT database. And so that's what Centrifuge was developed for. Centrifuge was developed to be able to have all of that information in memory. And so what we suggest to people instead is that they make their own databases, that they make their own Kraken databases and put whatever they feel they need and whatever they want in that database. And we just provide them with the simple, I would say, scripts to download them. So we're currently updating everything from Kraken, and I will say that I am promising a new mini Kraken in the next 2 weeks or so. And then we also updated what the standard Kraken database is. And so if people just download the standard Kraken database, it will include everything from RefSeq, from bacteria, archaea, and virus.
Cool. So I haven't heard about Centrifuge before. Is it a separate tool?
Yeah, so Centrifuge is a separate tool. It's actually written by a couple of one postdoc in the lab, another who used to be a postdoc in the lab, Daewon Kim, I think, is the first author. Florian Breitwieser is another author, and Lee Song is a PhD student in the lab, and he's the last author on the paper. And the three of them worked together to create Centrifuge, which does similar classification, but it uses a very different database. And so it uses an FM index instead of k-mers. I think that Kraken and Centrifuge have different pros and cons to them. And I think that we actually use both whenever we're running any sample. But I think that definitely the biggest pro, I would say, for Centrifuge, if someone wanted to use that instead, is if you need a large database. Because Centrifuge has the ability to hold the whole NT database in it as what it's comparing against for a sample.
Mm-hmm. And what is its downside when compared to Kraken? So, like, when would you use Kraken instead of Centrifuge?
I think something that we've noticed is that when it comes, my postdoc Florian is going to be not happy with me to say this. So I think the first thing to note is that we actually, for a number of our samples, we prefer not to start out by running everything against the NT database because it ends up with a lot of false positives just by the nature of the database itself, not by the nature of Centrifuge as a program. I think that I would say it's been easier for me and it's more comfortable for me to create a Kraken database and run samples through Kraken and analyze samples using the Kraken output rather than to try to use Centrifuge. And so Centrifuge is really useful, but I think that anytime we've run it, it's given false positives, not because the program itself is faulty, just because it's using such a large database by default. I don't know if it used the same database, what the differences would be, because I've never tried to do so.
Okay, got it. But how do you know those were false positives? Because in your sample, presumably there may be some contamination. There could be anything in there. And so how do you decide what to include in your Kraken database?
The samples that we were using that I was running were cornea samples that we have, where we know what they are, where we know like what the infection actually is. And so a lot of what I've been doing lately is trying to make a database of eukaryotic pathogens which is accurate. Because when we started doing cornea infections and analyzing cornea infections, we found that a number of the pathogens that infect corneas are not actually complete genomes at the moment. So what I was actually testing samples from corneas that a collaborator of ours had. These are not from any patients, but they have very specific infections. And so when we run them against Centrifuge, it gives us so much information about everything that it thinks that's in that sample, to the point where it's not necessarily useful because the database is so large and there's a number of false positives. So what we have also found in terms of what's in those databases is that there's contamination in the samples, sure, but there's also contamination in the sequences themselves. And so even I would claim that a number of the genomes in the NT database have human contamination in them. So there's human DNA in those genomes. And I think that that's where a lot of our false positives stem from. Because then if something gets classified at a very specific level at one of those genomes. It will choose that genome over human. And what we don't want to consider any classification where it might potentially be human. Because we are not, in a lot of our samples, we don't care about the human DNA, we care more about the microbial DNA.
Okay, interesting. Is there anything you would like to share with the audience, maybe about your future plans? What are you working on?
Yeah, so I guess I'll put this out there. I'm actually very soon going to be releasing a eukaryotic pathogen database, which we have cleaned through a number of cleaning steps. And this is meant to be a database where people with samples that are potentially eukaryotic pathogens, where they can run it against it and be confident in what they find. And so previously, these were all draft genomes. These are 250 eukaryotic pathogens genomes, but they were all in draft form because they have not yet been completed. As people in the field know, draft genomes just have a lot more contamination. And so what we decided to do is, instead of waiting for the various different labs to complete these genomes and finish removing any contamination and combining them into one single genomic sequence, what we wanted to do is clean up these genomes. And so we've actually just finished working on that, and the paper is in process of being written and will be published soon. And also in terms of the software side, I do want to say that we are updating Bracken for all of the Bracken users out there. And Mini Bracken, we will have a new one in the next couple of weeks. And Bracken version 1 is currently out there on GitHub.
Cool. Well, Jennifer, it was nice to talk to you.
It was great talking with you too.